Having discussed the antigen and the antibodies individually, now let us go on to their reactions. So we now study antigen-antibody reactions, which are of two types: precipitation and agglutination. So as I've told you, this is an interaction of an antigen-antibody, antigen-antibody, but what type of antigen? So for precipitation reactions, we have a soluble antigen, and for agglutination reactions, we have a particulate antigen. Remember, P is not for P, my favorite mnemonic which I use everywhere in path and in micro. So P is not for P. For precipitation, will you add particulate antigen? No, you will add soluble antigen. And for agglutination, you will measure the particulate antigen. So let's start one by one. I'm starting with precipitation reactions, and they can either occur in a liquid media or they can occur in a gel. So I'll first take up the ones that are occurring in a liquid media, and the first one is when antigen and antibody will meet, it will form a ring. The second one is that when the antigen and antibody will meet, it will form chota floccules. So when I say that an antigen over here and an antibody over here, they are gonna meet in this zone, that is where a ring is formed. Can you see a ring over here? So that becomes a ring precipitation reaction. How simple is it? There's an antigen, there's an antibody, they have combined, they have made a ring, and I get a ring test. I mean, nothing could be as easy as this. So is there any test that you've studied? Yes. We have studied in bacteriology, A for A, there is an Ascoli's ring precipitant test for anthrax, and there is a streptococcal Lancefield grouping. I hope you remember from your streptococcus chapter that streptococcus Lancefield grouping is for the beta hemolytic streptococcus. And which antigen am I talking about? We are talking about the C antigen, the carbohydrate antigen is what we are speaking about. So remember, the streptococcal Lancefield grouping for, of course, streptococcus, and Ascoli's test for anthrax, both of them are going to result in the formation of a ring. They are ring precipitation reactions. Is there anything that I have to be a little cautious about? Yes, there is a hypothesis known as the Marek's hypothesis. What is Marek's hypothesis? It says that when your antigen and your antibody are going to be almost similar in amount, that is when a ring will form. So can I say that this becomes the ZOE? This becomes the zone of equivalence. When the antigen and antibody are in the same amount, you can see that the precipitant formed is at its maximum best. Means anything less or more is going to create a problem. So if there is excess of antibody, do you see any precipitant forming? No. If there is excess of antigen, do you see any precipitant forming? No. So what are these known as? When there is excess of antibody, this is known as the prozone phenomenon. A lot of students ask me, what is prozone phenomenon? That is when antibodies are in excess. Lattice nahi banega. The lattice, the antigen-antibody reaction will not occur. When antigen is in excess, that is known as the post-zone phenomenon. That is known as post-zone phenomenon. So now let me show you another picture of how it's going to come. So look at this. When they'll say that the antigen and the antibody have become equal in amount, you will say jab equal hai, that means this is the zone of equivalence. The lattice has formed so beautifully, you will see the ring. If I on the other hand say that there is too much of antibody in the sample, there is way too much of antibody, that is what I have said. If antibody is in excess, what phenomena is it? It's a prozone phenomena. On the other hand, if I say that there is excess of antigen that is present, if there is too much of antigen that is present in the situation, then you say this is a post zone. Many students get confused. Pro and post mein kya hoga? So dekho, pro sounds earlier, post sounds later. Antibody and antigen में से B comes earlier and G comes later. ठीक है? So when antibody is in excess, it is a prozone phenomenon. When antigen is in excess, it is a postzone phenomenon. Go in an alphabetical order. This concept was known as Marek's hypothesis. Now what have we done? In liquid, I have already finished the ring test. I'm going to go on to the flocculation test. So what are the flocculation tests that you have to know? That now instead of making a ring, whatever antigen and antibody will form, that will basically form a floccule. So either that could be you could see the floccules on a slide, or you could see the floccules in a tube. So देखो, Tube is known as the Cahen test and slide is known as the VDRL test. But both of them are for the same thing. Slide and tube, ye VDRL and Cahen, what are they for? They are for Treponema pallidum, basically syphilis. They are for Treponema pallidum, that is going to be syphilis. But what we use more commonly is of course going to be the VDRL. So please remember the VDRL is very famous question. VDRL is which kind of immunology reaction? It is a slide flocculation test. It is a slide flocculation test, as it is Ratlo, slide flocculation test. So coming back, what all have we studied up till now? I told you that we had to do liquid media. Either you made a ring in that antigen antibody ke liye. Ring mein, do a quick summary with me. Ring mein you had two things. Either you made an Ascoli's ring precipitin test for which organism? A for anthrax. Or you did a streptococcal Lancefield grouping. So these were the two that you had put under the ring precipitation. For flocculation, you said, ma'am, you could make slide flocculation, which example you gave was VDRL. And you could do a tube flocculation where you gave an example of the Cahen test. So these two are certainly done, but you were doing all of this in a liquid. Now it's time to change into a gel. Gel means what? Agarose gel. So you're going to use a 1% agarose gel over here. And now comes the real thing. I've seen most students find a problem here. So listen to me carefully. Precipitation reactions that happen in gel media. Before I go on to those, you need to understand two definitions. What is diffusion and what is dimension? So if I tell you, I have done the whatever test I'm doing, I have done it in a test tube. If I say I've done it in a test tube, you'll say, ma'am, one dimension mein kara hai. If I say I have performed it on a slide, you will say you have done it on 2 dimensions. How do I learn? So the way you write test tube, it looks like one dimension. The way you write slide, isko flip karo, it looks like a 2. So simple. Test tube means 1 dimension. Slide means 2 dimension. Many people also learn कि test tube ऐसा 1 की तरह बनता है and slide has length and breadth. तो slide में 2 dimensions and test tube में 1 long dimension. Simple. Test tube 1 dimension, slide 2 dimension. Dimension का story tick mark. What is diffusion? Single diffusion. What is single diffusion? When only one thing is moving. When only the antigen, for example, this is an antigen and an antibody. If only antigen is moving, one person is diffusing. So this is single diffusion. If antigen and antibody both start moving, that is then known as double diffusion. So remember, if one is moving, single diffusion. If both are moving, double diffusion. You've understood the meanings of diffusions and dimensions. Let's do a very quick recap. Test tube matlab which dimension? 1 dimension. Slide matlab 2 dimension. If only antigen is diffusing, single diffusion. If antigen antibody both are diffusing, double diffusion. Now you must have heard of a lot of tests, but I know there are 4 tests. ठीक है, दोनों उनका नाम याद करना is like humanly impossible. They're such difficult ones. But the purpose of my lectures is to make those humanly impossible things a little possible for you. So what is that OOTD? Now you're all Gen Z people, guys. You know what is OOTD? Outfit of the day. So I'm going to use your Gen Z Instagram slangs and hashtags into this particular slide. But now I know that as medicos, our life does not revolve around OOTDs. Unfortunately, as medicos, we are so much into studies. Our life is more into topic of the day, mnemonic of the day, image of the day. I also keep having these series on, on social media, IOTD, TOTD or MOTD, and it's quite sad that where our counterparts and colleagues who are non-medicos are doing OOTD, we are in an altogether different world. So today we will also do OOTD, but not outfit of the day. I'm going to use a tube. So tell me, tube is what dimension? Tube, T1, single long tube. So you are basically talking about 1 dimension, 1D. So when you're having a tube and you have 1 dimension, now you will say, ma'am, dimension to fix ho gaya. You are doing the test in 1 dimension. Now you can play with the diffusion. Either you're going to have a single O, so matlab you are going to have a single diffusion in 1 dimension. Either you're going to play with the test where there is single diffusion in 1 dimension, or you're going to play with the test where there is going to be double diffusion in 1 dimension. So simple, 1 dimension to fix ho gaya. Test is being done in a test tube. Single diffusion means only antigen is moving. Double diffusion means antigen and antibody both are moving. So when it is single diffusion, it is known as the Auden test. It is known as the Auden test. The O for Auden test. But when it is double diffusion, so ma'am, double words aana chahiye. That is known as the Oakley-Fulthorpe test. That is known as the Oakley-Fulthorpe test. I'll repeat it. Now if I write this in the form of a sentence, so what they'll do in the exam, they will say, can you tell the meaning of what is single diffusion in one dimension and what is double diffusion in one dimension known as? And you'll be like, okay, hang on. As soon as I know that they're talking about one dimension, you'll say, arey, one dimension matlab they're talking about the test tube. They've asked me about the OOTD mnemonic. So if they've asked me single diffusion in one dimension, I will mark as Auden test. If they've said double diffusion in one dimension, I will mark Oakley-Fullthorpe test. Have a look at the pictures now. See, it's in front of you. Single diffusion in one dimension, double diffusion in one dimension. One dimension matlab the test tubes are in front of me, correct? Single diffusion means who is moving? Only the antigen is seen moving. Only antigen is moving. This means this is the Auden test. And if I see the antigen and the antibody both are moving, that means it is double diffusion. That is going to be Oakley-Fullthorpe test. Simple. If you see only antigen moving, Auden. Antigen and antibody dono move kar rahe hain, do words use kar lo, Oakley-Fullthorpe test. In case you don't want to put so much of logic into it, Auden is sounding like ek din. Everything is ek. One diffusion, one dimension, one day. Okay, one diffusion, one dimension, one day. So that is Auden, and the other one becomes a double diffusion in one dimension. So now you're done with the one dimension. You're done with the test tube wala thing. So now whatever is left is going to be the two dimension, means slide wala thing. Okay, so under this, the first one that you have, you'll say, ma'am, now we are going on to two dimension. Two dimension means now you're going to deal with a slide. And अब फिर से वही है. Now you're going to talk about the diffusion. The diffusion can either be single diffusion or double diffusion. We'll start with single diffusion. Single diffusion is known as radial immunodiffusion. That is going to be RID. RID is going to have a radial immunodiffusion. So if you read the word, I'm going to pick up the same alphabets. Radial immunodiffusion. Radial matlab, I'm going to see a picture which is having a circle, because circle has a radius, hai na? So I'll see a radius wala photo, and if I pick up the eye, it's looking like 1. So it is what diffusion? Instead of immunodiffusion, can I say it is one diffusion? And ऐसे slide में हो रहा है, तो it has to be a two dimension. So now you know कि ठीक है, two dimension तो slide you know. You just wanted to know single diffusion or double diffusion. So you'll say अच्छा radio immunodiffusion में तो one diffusion word is anyway coming. So it becomes a single diffusion. Means who is going to move? Single diffusion means only the antigen is going to move. So let's have a look at it. What did they do over here? They took a gel, or basically you said now all of these are gel based. So they took an agar gel, and in that agar gel they have already added an antibody. After that, there are certain wells that are present in which they are going to add the antigens. And who is going to move? Only the antigen is going to move. Single diffusion है ना, so only one person can move. Only the antigen is going to move. तो antigen move करेगा. Once it will meet the antibody, there is going to be a reaction. So you can have a look at this. You can see over here that as there were rings formed, or you can say there were precipitin arcs formed, precipitation reactions हुआ, antigen antibody reaction हुआ, but we are even measuring the diameter. Every ring that is formed, I am measuring the diameter. So this is just not to know antigen antibody reaction. This is also going to give information about the concentration and the subclasses. It's also going to give concentration and subclasses of different antigens and antibodies can be given over here. Please note, this is also known as, only for INI-CT I'm telling you, this is also known as the Mancini's method. This is also referred to as the Mancini's method. So I'm repeating once again, radial immunodiffusion. Radius will remind you कि ये कुछ slide में हुआ है. Slide मतलब it's a two dimension, and immunodiffusion मतलब one diffusion. It has occurred in single diffusion. So what am I left with now? Now I'm left with DDDD. Now ma'am, you are left with two dimension and double diffusion. What did I say? Two dimension and double diffusion. Is it sounding like D, D and two D's over here? DD. Does it remind you of any cartoon? Remember Dexter? Have you guys seen Dexter cartoon? Dexter ki ek sister hai, that is DD. Hai na? So when DD comes into Dexter's laboratory, can Dexter work? No, Dexter is like too panicky, and DD breaks everything in his lab, all his test tubes and experiments. So how I have learnt it is that when DD will come in the laboratory, please note that this is gonna be ouch. Everything is going to break. DD is going to break everything. So double diffusion in 2 dimension, D is known as the outer Loney test. Ouch. DD is going to be outer Loney test. And why did I say DD? Because ye to DD to is telling you double diffusion 2 dimension, DD. And I've told you that DD has come to ouch hoga, that is outer Loney test. But why did I write EEDD? Because an example of the outer Loney test is the LX gel precipitation test that you do for Corynebacterium diphtheria. So now everyone write DD with me, which basically tells you that when you write DD is coming, firstly you will say that ma'am, all the D's have to come. This has to be a double diffusion in 2 dimensions. Then when you're saying DD is coming, so ekdam se I'll be like, okay, ouch, everything is going to break. This is the outer Loney test. After that, you are left with the E's. So now I know we are dealing with the LX gel precipitation test over here. Let me come back and show you the picture. I am saying it is a double diffusion. Everyone is going to diffuse. Antigen will also diffuse. Antibody will also diffuse. And I can see over here, I have kept an agar plate with a lot of antibodies. Okay, and in these wells, if you look, I have added antigen number 1, then I have added antigen 2, I have added antigen 3, I have 4, 5, different antigens I have added. Why? Because I realized that all of them start coming out. Antibody ke response mein, all of them start diffusing. Sometimes I will see that these, like this antigen from here and this antigen from here, they were moving and they ended up making an arc. This antigen from here and this from here, they ended up making a continuous arc. Matlab this and this antigen is the same. When you go and hug someone, you're best friends with them. You are very similar. That is when you go and hug someone. So if the arcs have hugged each other, this means this and this antigen is identical. But if you go and fight with someone, if you go and have these intersecting line, matlab ye aur ye antigen same nahi the. They were non-identical. And on the other hand, you kind of give a formal hug. Kind of hug, kind of war. Kind of hug, kind of war. Matlab partial identity. That is known as spur. So repeating once again, if you get a precipitin line which is a common arc, matlab they are identical. If you get as a cross, matlab the antigens are non-identical. And if you get a kind of cut and kind of meet, what did I say? Kind of cut and kind of meet, that is a spur formation, that is a partially identical. So let me ask you, yahan pe kya dikh raha hai? What do you want to take this? Yahan pe what kind of an arc is forming? You will say, ma'am, mujhe to this is looking like a continuous arc. What do you want to say? Are these antigens in these wells, are they identical or non-identical? Continuous ho raha hai, so they are identical, right? If they would have been cutting like this, they would have been non-identical. And if they would have been like partially cutting like this spur, they would have been partially identical. This is known as the DDDD test, outer Loney test, LX gel precipitation test. And that means that on that note, you have finished finally with all of your gel precipitation tests are over. Yes, everyone's okay with this. Now what do we move on to further? Now you say, ma'am, isme to bahut zyada time lag raha hai. Before that, I want all of you that please do a quick revision with me, because many students are unable to, memorize this only. So let me start. When I say, first I'll start with what? I'll start with OOTD. What is OOTD? Means I'm using a test tube. A test tube means I'm using a one dimension. So single O means I can have a single diffusion. What do you want to call single diffusion? 1, 1, 1, 1. You want to call it an Auden test. What do you want to call the double diffusion? Double diffusion you want to call as the, yes, the two names should come, ma'am. That is Oakley-Fulthorpe test. Okay, Oakley-Fulthorpe test. This was about the one dimension story. Now tell me about the very easy, the two dimension story. So two dimension may be same. Either you're gonna have a single diffusion in 2 dimensions, or you're gonna have a double diffusion in 2 dimensions. So tell me, if you have a single diffusion, so 2 dimension kya hua? Slide. Ma'am, this will also occur on a slide. This will also occur on a slide. So single diffusion matlab radial immunodiffusion, Mancini method, immunodiffusion, one diffusion, whereas double diffusion in 2 dimensions is going to be ouch, that is outer Loney test. Simple. Auden, Oakley-Fulthorpe, RID and outer Loney. All of these were happening very slowly. Ma'am, kuch electric current lagao, make it electric. All these reactions should happen jaldi. So what I'll do, I'll take the same agar plate. You will see I have added a lot of antigens over here. This is an antigen that has been added over here. and in this trough above I am going to add an antibody. तो यहाँ पर है antibody, यहाँ पर है antigen, but what have I done? I have applied an electric current. I have applied an electric current and I have separated the antigen into many parts so that now proper lines will form. Proper lines will form whenever the antigen antibody will meet, proper lines will form. I have used electric current. So why do I want to call it immunodiffusion? No, now I'll call it electroimmunodiffusion. Because what have I done? I told you na, every antigen is very complex. So मैंने इसको छोटा parts में, like you say antigen antibody reactions is a very complex chapter. I'll say okay, never mind, I'll break it into small topics. So by applying electric current, I have broken it into small antigens, and that is what you've done. So I'll repeat this once again. What do? You take an antigen to begin with, you apply an electric current. After that those antigens get separated. अब पूरा छोटा topics हो गया, all छोटा antigens are ready. Now you're gonna add antibody over here. So these antibody and these antigen will meet and jaldi they will give you precipitin line. So this is a faster method. This is electroimmunodiffusion. You say ma'am और भी जल्दी कर दो इसको. Okay, I'll say अभी तक what was happening in the previous one was only the antigen was being separated like this and only one thing was moving, only antibody was moving. अब दोनों को move करा देते. Means if I place an antigen here and I place an antibody here and I do something like counter immunoelectrophoresis, means I create a kind of an electric charge that both of them start moving towards each other. It is a mutual attraction. Don't you think the line that will form on their junction will be even faster? So this is definitely going to be faster. Repeating, in electroimmunodiffusion, एक side का current था, only the antibody was going. Over here, antigen antibody, both are going to move. So this is counter immunoelectrophoresis, which is going to be much faster. But none of them are telling me the quantity of the antigen or the quantity of anything. For quantitative assessment of the antigen, I am doing rocket. Easiest to identify, rocket immunoelectrophoresis. You will see so many rockets in this journey. So what will you do? You will keep the antigen in the increasing concentrations, and you will add the antibody in the agarose gel, and you will be able to see that as your antigen concentration is increasing, you will realize that the rockets are also increasing. So this helps you in doing the quantitative assessment. So remember, when we talk about rocket electrophoresis, the rocket is going to help you in the quantitative estimation. Guys, that finally wraps up all of your precipitation reactions. Liquid wala ho gaya, gel wala ho gaya. If you want to add a little bit of electric current, you can add electric current only in one dimension, matlab in one way, electroimmunodiffusion. दोनों तरफ से करना है movement, तो counter immunoelectrophoresis, and if you want to quantify, you do rocket electrophoresis. Done with that, and we move on to the agglutination reactions, which are said to be better than precipitation reactions. Now first tell me, it's going to be a reaction of antigen and antibody, but which antigen? Soluble or particulate? Remember, P is not for P. Precipitation mein I did not have a particulate antigen, but here can I have a particulate antigen? Yes, I can. There are two tests, direct agglutination, indirect agglutination. I'll start from here. Direct agglutination, I'll show you one diagram. ये देखो, direct मतलब मैम direct, antigen है, antibody है, agglutinate करो, खत्म, direct. What is direct agglutination test? You have a particulate antigen, you add the antibody, हो गया agglutination. That is direct agglutination test. Either you can do it on a slide, or you can do it on a tube, or you can look at it under the microscope. If you do it on a slide, you must have seen वो जितना भी A blood group, B blood group वाला photos that I show you in pathology, they are all slide agglutination. So on the slide you are seeing अच्छा like over here A positive है, B positive. So you're doing it on a slide. That is slide agglutination. Tube agglutination will obviously happen in a test tube. What all comes? Can you tell me the names? Vidal test. Vidal test for what? For typhoid, of course. Okay, then what do you know the Weil-Felix test for? That is for rickettsia. What is the Paul Bunnell test for? It is for Epstein-Barr virus. What is this SAT and CAT? So now you will say that ma'am, SAT and CAT are being done in a tube. So you told me, I'll tell you what is SAT and CAT. So you told me, ma'am, ya to this will happen on a slide, or this is going to happen in a test tube, Or you said there is a third version of it. Slide and test tube is okay. You said there is a third version that is gonna happen in the microscope. So what is it, the third version? वो भी हमें बता दो. So the third version, that is microscopic agglutination testing, is going to be a mnemonic or is going to be an example called MAT. So now you will obviously ask me, ma'am, what is this CAT, SAT, MAT that you're doing? Here you did SAT and CAT for tube agglutination. Here you're doing MAT for microscopic. So let me tell you, what is this CAT, SAT, MAT? पहले तो full form करो. Full form is their agglutination tests. Na? So AT always has to stand for AT agglutination test. ठीक है, तो CAT is cold agglutination test. STAT is going to be standard agglutination test. And these are done in test tubes now, because M for microscopic agglutination test. This is the one that is done under a microscope. So simple. Cat sat on a mat. That is how I have learnt it. Why? Cat sat on a mat. Who is cat? Cat is my billy. Cat is a billy, right? So you will say these are for organisms, my billy. Every time you get this question in the paper, you will say cat sat on a mat, and cat sat on a mat matlab you are talking about my billy, means the cat is done for mycoplasma. Cold agglutination test is for mycoplasma. Standard agglutination test is done for brucella. And finally, the microscopic agglutination test is done for leptospira. And I remember just 48 hours before the NEET PG 2023, we had discussed it on the YouTube channel on Prep Ladder, and I remember this came up in the paper. We did the same mnemonic. The only one thing we discussed before the exam was this: cat sat on the mat, my billy. So chalo, we said ke chalo, let's go by gut feeling, and this came up in the paper. Of course, students were very, very excited. And this is a mnemonic which is tough. Examiner doesn't know हमने mnemonic बनाया है ना. Examiner is thinking they're all sounding the same. So let me confuse, but now we know the mnemonic, right? So cat sat on the mat, and that is mycoplasma, brucella, leptospira. And now you know what reaction are they? They are anyway cat sat mat में तो पता ही है they are agglutination reactions. You should be knowing microscopic वाला तो is obviously under the microscope, but the other ones are going to be tube agglutination. So slide में there is only blood grouping. Microscopic में there is only microscopic agglutination test. Everything comes under tube agglutination. So what? Vidal for typhoid, Wheel Felix for rickettsia, Paul Bunnell for EBV, cat sat mat सब हो गया. अब क्या बचा? Coombs test. Okay, so what do you need to know about Coombs test? There is a direct Coombs test that you have, and there is an indirect Coombs test that you have. So please note, very commonly you will be doing this direct and indirect Coombs test obstetrics and gynecology. So for example, you have direct में तो कुछ ऐसा बिल्कुल direct होगा ना, 1, 2 खत्म, always a direct test. So what have you done? Let's assume I have taken the fetal blood sample over here, and I am saying the fetal cells are already coated with antibodies. The fetal cells are already coated with antibodies. Why? Because maybe this is a case of erythroblastosis fetalis. This is a case of erythroblastosis fetalis. So they say that the fetal RBCs are already coated with antibodies, but they are not agglutinating. They are incomplete antibodies. So you please add something. Coombs test में क्या add करोगे? Coombs reagent add karoge. You're always gonna add Coombs reagent. What is Coombs reagent? Anti-human globulin. You add anti-human globulin. So whatever gap was there in between them ना, वो anti-human globulin ने जाके पूरा gap ऐसे bridge कर दिया. So now will you be able to see that agglutination very, very easily? Yes, you will see an agglutination in this. So you will say this agglutination will only be seen if the fetal cells already had antibodies on their surface. Let's do a very, very quick recap. I'm talking about the direct Coombs over here. I have assumed that my fetus over here had erythroblastosis fetalis. So the fetal cells already had covered themselves with some antibodies while in utero. So when I have taken and added another antibody, so it has gone and bridged the gap. I have seen an agglutination. I will say DCT, direct Coombs test, is positive. The fetus had antibodies. Now, if I want to do the indirect Coombs test I am doing when I want to find out whether the mother has antibodies in the serum. So over here, I have only taken antibody serum, matlab I have taken the mother's serum. I am assuming the mother has antibodies in her serum. You will say, ma'am, ab kaise karaye test? Antibody to hai. So now I will have to indirect hai na, I will have, where are the cells? पहले वाले में तो you took the cells, so you just had to add one antibody, direct था, खत्म. यहाँ पर तो you've taken antibodies, so you will now add some test red blood cells also, and you will add the anti-human globulin also. So simple, in a direct Coombs test, all that you had to add was one anti-human globulin, khatam. But in an indirect Coombs test, because you have taken the serum, you will have to add the RBCs also and the anti-human globulin also. Then if by chance antibodies are there in the mother's serum, then all these things together will make an agglutination. So if you get an agglutination or you get an indirect Coombs test positive, that means the mother had antibodies in her serum. So if you want to detect the fetal antibodies, if you want to detect fetus ka problem, it is direct. If you want to test problems in the mother, that is going to be indirect. The same Coombs test you're also using for what? For autoimmune hemolytic anemias. Remember, I have told you in autoimmune hemolytic anemias in path also you do the Coombs test only. So these were all examples of your direct agglutination. Direct agglutination, ya to you did it in a slide or on a tube or on a microscope. Direct antigen liya, antibody liya, khatam. Provided the antigen was what? For agglutination reactions, P wala antigen, particulate antigen. What if you say, ma'am, I have a soluble antigen? Can you do an agglutination reaction? No, you will have to make it particulate. How will you make it particulate? First you make it particulate, then you do the reaction. So this is an indirect agglutination test. How do I make it particulate? So for indirect agglutination test, I will have to make that antigen to aise akele ghum raha tha, soluble antigen tha. If it is a soluble antigen, I will have to basically absorb it on either a red blood cell or I can even use latex particles. I will either use latex. So for example, if these are latex particles, all these antigens that you have, they are going to come and absorbed over here. So now it has become a particulate antigen. Now if it has become a particulate antigen, I just have to add an antibody and the reaction shall occur. So look at that slide. You have a soluble antigen. Pehle you stuck it onto some latex or something. Then you added an antibody and then you got a reaction. So simple. Direct mein to kya tha? Antibody, antigen, direct. Chalo khatam. But indirect mein first you convert it into particulate and then you do the reaction. And one very important test is latex agglutination reaction. Latex agglutination test, for example, can be used for anti-streptolysine O antibodies. मतलब streptococcus infection के लिए we are doing latex agglutination testing. Okay with everyone, so your precipitation agglutination reactions are done. I know it's tough. One quick 2x की speed पर you will need a revision. That is a must. Now going forward, talking about the complement fixation test. Complement fixation test, you just need to know one principle and two examples. पहले examples याद कर लो, the Wassermann test and the Wassermann test and Treponema pallidum immobilization test. Treponema pallidum immobilization test and the Wassermann test is also for Treponema pallidum by the way. So ये दोनों जो Treponema pallidum tests है, they are complement fixation tests, but out of them Wassermann test is तो no longer done. Treponema pallidum immobilization test is done. It's in fact said to be one of the most specific tests that you have for Treponema pallidum. So in fact, what is happening? देखो नाम बताओ. Complement. Ma'am, you will need some complement and you are going to fix the complement. Complement fixation test. You are going to basically a complement है उसे busy कर लो. How do I make it busy? I will have an antigen. I will have an antibody. Now tell me something. If an antigen and antibody will combine, what do? Don't they go and fix the complement? That is what we always study. If an antigen and antibody are there, they will always go and make a complex with a complement. So antigen, antibody or complement to busy ho gaya. Will this complement ever have time to kill the red blood cells? Kill the sheep red blood cells? No. So can I say that if the antigen and antibody are present, that means the complement has been made busy. Complement has been fixed. And fixed matlab relationship mein fix ho gaya. Rishta pakka types. Relationship fixed. Which basically means there is no complement for RBC lysis. So there is no RBC lysis. So now it's very clear that if the RBCs are not lysed, this means patient ke paas antibody tha. And if the RBCs would have been lysed, this means that patient ke paas antibody nahi tha. So now very simple way of learning it, ulta chalega. If the RBCs are looking intact, matlab RBCs have not lysed, this means that the patient has antibodies. That is a complement fixation test. Nowadays we do Treponema pallidum immobilization as a complement fixation test. The next type of antigen-antibody reaction is a neutralization reaction. Neutralization means if you have a toxin, neutralize the toxin, add antitoxin, right? So like you say that if someone has been bitten by a snake, you give antivenom. Same way, if there is a bacteria with a toxin, you give antitoxin और वो हो जाएगा neutralize. So what are the two tests which are going to be coming under this? There is a Schick test. Schick test is done for what, guys? Schick test is basically done for Corynebacterium diphtheria. It is a neutralization test. Negler's reaction, Clostridium perfringens, that is also a neutralization. You might not understand these right now. I can totally understand that because you have not read Corynebacterium diphtheria or Clostridium perfringens. So I don't want to repeat it. Abhi padh ke fayda nahi hai. You go to that chapter, come back to this after that. It will be crystal clear. Then you will finish this chapter in seconds. Okay, all this is done. Now we move on to some machines. What is ELISA? What is a card test? What is fluorescence test? So now what we start with next is going to be ELISA. First, you need to know that this is an ELISA plate, what you have in front of you. This is an ELISA plate under which you can see there are 12 wells in this direction and there are 8 wells in this direction. So total becomes 12 into 8 and that is going to be 96 wells. This could be a question in path, micro or biochem, wherever ELISA is used. Okay? So 12 and 8, total 96 wells, 96 testings can be done at the same time. Such a useful thing. But what is ELISA? Enzyme-linked immunosorbent assay. So means it's an assay, it's a test in which I'm doing some immuno-antigen antibody stuff. But that is with the help of an enzyme. So first I want you to ratta lagao 3 things. Then I'll tell you the types of ELISA. In this, if you ask me what is the enzyme is HRP. If you want to know the full form, horse radish peroxidase. It is going to be horse radish peroxidase. Peroxidase tells me that yeah, it is an enzyme. Then you have a substrate called hydrogen peroxide, H2O2. Enzyme will always break something. Na? So wo substrate kya hai? That is H2O2. And in the end, how will you get to know test is positive or negative? You should have some color. So the color is some form of benzidine that is used. Over here they are using tetramethyl benzidine. So 2-3 things you ratto. Which is the enzyme? Peroxidase. What is it going to break? H2O2. And what is the color? There is a color known as benzidine. So now what am I going to tell you? There are 4 types of ELISA that we must know for the exam, which I'm sure all of you know. Direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA. Now let's start one by one. Direct में क्या measure कर रहे हो? पहले तो रट लो. Direct में what are you measuring? Direct में you're measuring the antigen. Sandwich में you're measuring the antigen. Competitive में you're measuring the antigen. Means where are you testing the antibody in the indirect ELISA? Point number 1. Up detail में चलते. You should know now what am I trying to look for in the patient. So in all of them, you are trying to look for antigens in the patient. The only one where you're trying to measure antibodies in the patient is indirect ELISA. Now let's go into the details. So what is a direct ELISA? Starting from the first one. So you will say, sub mein ma'am, it is antigen detection. This means this is that ELISA well. If I want to test for antigen, this means if I want to test for the antigen, this means the first thing that I'll add is the patient sample. I want to find out antigen in his sample, na? So I will add his sample first. Then patient ka kaam to khatam. He has given me his antigen. Now I will add an antibody in the lab. You know what I'll do? Smartness that I'll do. I will combine that antibody with an enzyme. Which enzyme are we dealing with? Horseradish peroxidase. So for example, let's assume patient mein antigen tha. Okay? If the patient had the antigen, if I'll add an antibody with an enzyme, will an antigen-antibody reaction occur? Yes. Antigen-antibody reaction hoga. How will I get to know? You say, ma'am, give the substrate for this enzyme, add the substrate for this enzyme, that is H2O2. This enzyme will then go and break the substrate, add some color. You will get to know what color. You add benzidine and you will get a color positive. So simple. If there is an antigen in the patient's blood and you have an antibody add, the antibody will get added to it, get attached to it. If you add an enzyme, the enzyme will break this H2O2. H2O2 will change the color and the color of benzidine will come out. So simple. Antigen hai, to color aayega. Means I can even measure the intensity. How much of color is coming will tell me how much of antigen is coming. That is, so you must have heard that reports always give you quantity. Antigen is 4 ml, 5 ml, 3 microliter. How? Because that's on the basis of the color. More color matlab there was more antigen that was present in the well. So simple. Now you know if you want to test an antigen, that is direct ELISA. But what is indirect ELISA? Indirect ELISA mein what do you want to test, guys? Indirect ELISA mein you are gonna basically measure the antibody. You are gonna measure the antibody in the patient. So now you will say that ma'am niche to antigen dalna padega. So now you will add antigen on your own from your lab. You have these machines ka manufacturers, they'll give you antigen. Then you will add the antibody from the patient. You will add the antibody from the patient sample because you want to test whether does he have the antibody or not. So you'll say antigen ma'am I contributed on my own. Then I added the patient sample. Then to confirm this, I did the same process again. I added another antibody. This time I had conjugated that with an enzyme. That enzyme went and broke the substrate H2O2 that I added, and finally I got the color. So more the color that I get means more was the antibody in the patient's sample. So ultimately I realized adding an antibody which is linked with an enzyme and the enzyme will break the substrate H2O2 and the color will come out. That was there in direct ELISA also. That is there in indirect, that will be there in every ELISA. What are you detecting will change. In direct ELISA, direct patient ka sample dala, khatam. That's all that you had to do because you were testing antigen. Whereas in indirect ELISA, first you add an antigen, then you add the patient sample indirectly because now you want to test for antibodies. Let's come to the third one, sandwich. Ab sandwich banao. Sandwich kaisa banega? There are two slices of bread. And there has to be butter or some thing in between for a sandwich. So you will say ma'am sandwich matlab two slices of bread matlab antibody idhar, antibody idhar. There is antibody on both sides and you are basically having an antigen in the center. So ye ho gaya antibody and this is going to be an antigen in the center. So tell me, for everything you were trying to measure the antigen only na, so you will say ma'am patient mein to kya measure karna hai? Antigen measure karna hai. This means this antibody you will have to add and the upar wala antibody also you will have to add on your own. Patient say you're only going to get the antigen. So you add an antibody first, you capture the patient's antigen, then you add an antibody again, and after that the rest of the story is the same. Na? You add that antibody which has an enzyme is going to break the substrate H2O2, then the color is going to come. So sandwich ELISA is also going to be a variety of direct ELISA. Why? Because it is going to do antigen detection, but antigen ko humne sandwich bana diya. Either antibody, and then the rest of the story is the same. Coming to the last one, competitive ELISA. This was also for antigen detection, but here I am gonna create a competition. Competition means think of the topper of your class. He used to come to the class and say, "Arey, kuch nahi padha, I have not studied anything. I was slept off yesterday. I had gone for a movie." But what was he doing? He was actually sitting locked the hostel room and was studying or sitting in some library. So pehle se he has done his homework and come, hai na? So what am I doing over here? If you want to test for antigen, like you say that, ma'am, I want to test for free testosterone levels in a male patient. How much of testosterone does he have? So you want to test for some kind of antigen in a person. What will you do? Whatever antigen you had, usse pehle hi bahar. Finish the course in the library. Don't bring it to the classroom. Class mein aane se pehle those students who read and come na. So what have you done? You have taken the antigen from the patient and you have already added an antibody to it. So you have already done an antigen-antibody reaction outside the well. Alag se test tube mein kar diya. Pehle se course khatam. Now just imagine if you add this antigen-antibody mixture to an ELISA plate. And I know every ELISA plate I will be coating it with antigens. I have already coated it with antigen, but you'll say, ma'am, when that person has already finished his syllabus outside the class, why will he come and learn anything new? Even if you put him in the class, he's gonna chill. He will show everyone that, I don't study. Why? Because he has already finished his syllabus outside. So if antigen-antibody reaction as a competition has occurred outside, when it will come inside this well, it will keep floating like this. It will try to show chill, I'm floating around. I'm not going to show this reaction. Now when you do that test of yours, that is you used to add the secondary antibody and the enzyme and the color, all of that. So you will say that, ma'am, if for example there were 5 antigens in the beginning and you ended up adding 10 antibodies, assuming patient ke paas 5 antigens the and you had added 10 antibodies. So can you say 5 to consume ho gaye? How many antibodies were left extra? 5 antibodies were left extra. You ended up adding little more substrate, little more reagents. तो वो 5 antibodies में तो कुछ reaction दिखाया होगा, तो कुछ color तो आएगा. Little color will come. So now this works as an inverse. Competition में उल्टा चलेगा. If you get a lot of color, this means all your antibodies had basically gone free. Antigen था ही नहीं. So all these antibodies had the time in life to come and study. बाहर कुछ competition था ही नहीं, बाहर कोई पढ़ाई करा ही नहीं. That is why all the antibodies had come and they had gotten bound over here. That is why they gave so much of color. So if you get too much of color, this means बाहर homework हुआ ही नहीं था. If you start studying too much in the ELISA plate, if you start studying too much in the class, this means there was no homework that you did beforehand. On the other hand, if you stop getting any color, if you don't get any color, you will say, ma'am, there is no one studying inside this, no reaction happening inside this ELISA plate. This means all the reaction has occurred outside. This means the antigen was in excess. Now you are understanding the competition कि सब बाहर ही हो गया reaction. So simple, this works in an inverse relation, and the inverse relation is that if you are getting a lot of color in the end, this means the concentration of the antigen was low. And if you are not getting color in the end, this means the concentration of the antigen is high. So simple, if you are not studying in class, this means you have done your padhai bahar. And if you are studying too much in class, this means you have not done your padhai bahar. Okay, that is what is a competition. So now when we do a final summary, didn't I tell you direct, sandwich, competitive? In everything you study the antigen, but in everything, if you remember, more the color you get, more is the antigen. More the color you get, more is the antigen. But in competition, it is ulta. More the color you get, less is the antigen. And what were you doing in indirect? You were testing for antibody. Now the only difference that I'm going to do, this was all ELISA. There is something also known as CLIA. CLIA is known as chemiluminescence immunoassay. So firstly, what did I say? Immunoassay. Ma'am, ELISA may be immunoassay tha. But over here, you are using chemiluminescence. What does that mean? That means that you are not going to get a color in the end. You are going to get something shiny in the end. Abhi tak to you were saying color aaya, color nahi aaya. But now you are saying some chemical light is gonna come. If some light is coming, this means you have not used that benzidine. In ELISA, the color that you were using, was benzidine. You were using color, that is why you were getting color. But if you start using some luminal esters, you will start getting light signals. Baki sab same, antigen, antibody, platelet, sab same. Only difference is that in ELISA you are dealing with colors, and in chemiluminescence you are dealing with something luminescence, luminal, light, shine. That's the difference. Coming to the next, I'm sure all of you have also heard of immunofluorescence. What is it about immunofluorescence that you have to know? There is a direct immunofluorescence or direct fluorescent testing. Then there is an indirect fluorescent testing. So, sabke baare mein we are going to study. So, we will repeat it once again. Direct and indirect. Okay? Ab now, I tell you one, I teach it a little different in comparison to how others are taught it, because this one slide will give you everything. Lag to same raha hai, ma'am. It's looking antigen, antibody. In the end, you have added some light wala signal because fluorescence, everything is looking the same. Like I said, direct fluorescent testing is basically for testing the antigen. So now tell me, patient mein what are you trying to find? Antigen. So this is the patient ka sample that you have added. Patient ka antigen. What will you add on top of it? You will add an antibody and you will add some fluorochrome signal. You will add some fluorescent signal. Which is the fluorescent dye which is most commonly used for this process? It is FITC. Wherever they ask you fluorescent dye in any machine that is being used in Path, Micro and Biochem, fluorescent dye which a machine is using is always FITC. FITC stands for fluoroisothiocyanate. Fluoroisothiocyanate. So remember, in the patient you want to test for antigen, you add an antibody with the fluorochrome signal, reaction hoga, fluorescence aayega. So simple, if the antigen is present, the fluorescence is going to be present. Now this was direct fluorescent testing. What is indirect fluorescent testing? It is of two types. Indirect fluorescent testing mein you can test antibody or the antigen. You can test both and concept is same. If you want to test for antibody, What will you add from the patient? You will add antibody from the patient. If you want to test for antigen, you will add antigen from the patient. So the concept is the same. I just want to tell you that in direct fluorescent testing, you are directly measuring only antigen, whereas in indirect fluorescent testing, you can measure both antigen and antibody. And I'll give you an example. FTA absorption. Right now, you might have studied bacteriology, so you would know fluorescent treponema antibody. It's a treponema pallidum test and naam se hai fluorescent treponema antibody. So that is an example of the indirect immunofluorescence. Fluorescence word has come now. So direct for antigen, indirect for both antigen and antibody. Coming to the last test that we have in this chapter, and that is going to be the CART test. Of course, this is also an antigen antibody, and you can do CART test for so many things. I'm only giving you examples. You have CART test for hepatitis B, hepatitis C. There are CART tests for malaria. There, of course, how can I forget COVID? There are CART tests for even urine pregnancy test is a CART test. Why this blood? Urine pregnancy is also a CART test. So there are n number of CART tests. Typhoid, matlab typhoid fever ka bhi CART test hota hai. So it's an endless list. What I want to tell you is, they will ask you what is the principle of this CART test and how do you read it. Principle is, it is based on ICT, immunochromatography. It is based on ICT, which is immunochromatography. You will have a control line always has to be there. Please remember, the control line always has to be present, because this tells you that everything is going normal in your lab. Sab kuch theek chal raha hai. This tells you the validity of the test. So this has to be there. But when the test line will be present, when the test line, or you can say when the test line is seen, it is a positive reaction. When the test line is not seen, it is going to be a negative reaction. So what are these lines seen on? Ma'am, ye andar dekho, peep inside. What is this white color strip? Yahan se this white color strip is starting. Because I know that I will be adding my blood over here. How is my blood trickling like this and going up till here? How capillary action se kaise glide kar raha hai? Because inside what you have is a nitrocellulose membrane. You have a nitrocellulose membrane. So repeating once again, what are the questions you get? CART test is done for a million things, you know that. Principle, question number 1, 2019 NEET PG question. Principle immunochromatography, that was 2019. After COVID, so this becomes a million times more important. Immunochromatography, what is the membrane made up of? It is made up of a nitrocellulose membrane. What does control line tell you? It tells you the validity of the test. And test line to pata hai, if it is seen, it is positive. If it is not seen, it is negative. We are going to start our journey with number 1, the complement system, the first K. Now, I could have called it complement pathway also, but it's not one pathway, it's a system. It's a system of 3 pathways. So please remember, there is something known as the very, very famous classical pathway. There is something called classical pathway. Then there's an alternative route. So there is something called alternate pathway. Right? There's some alternate route of starting it, alternate pathway. And then there is something called MBL pathway, or lectin, or in other words, if you want the full form, mannose binding lectin pathway. Mannose binding lectin pathway. So simple, classical pathway, alternate pathway and MBL pathway. Let me tell you one thing, no matter whichever pathway you start, your end result is going to be a molecule called MAC. I'll tell you what is MAC. So first you will say that, okay, if everything is the same in between them, then why are there 3 pathways? Because their starting point, what are they activated by, that is going to make a difference. What are they activated by? So number 1, the classical pathway is activated by immune complexes. What do you understand by immune complexes? Immune complex means antigen antibody reaction. And which antibodies are going to come over here? IgM and IgG. Any immune complex that contains IgM and IgG will go and activate which pathway? The classical pathway. Talking about alternate, A for A, any immune complex containing IgA is going to go and activate the alternate pathway, A for A, simple. And when I talk about mannose, mannose is something in the bacteria. So any pathway which is directly activated by the microbial organisms, any pathway directly activated by the microbial organisms, that is going to be the mannose pathway. So simple, end result of everything is going to be the same. The starting is different. So classical by classical complexes, M and G complexes, alternate by A, IgA antibody complexes, and mannose by the bacteria or the microbial. Now let me show you what is the tough part going to be over here. So first and foremost, these are the three pathways which I've tried to put up over here. I'm going to tell you a shorter version of it, and then obviously I'll be telling you a detailed version of it. So let's start with the shorter version first. There is something known as the classical pathway. How do you activate the classical pathway? By immune complexes, by antigen antibody complexes. And can you see which are the set of molecules that you have activated first? The first molecule that I activated was C1. After that, I have a rule. First go for even numbers, then go for odd numbers. C1 will then activate all the even numbers, that is C4 and C2. They will get activated. After you have activated C4 and C2, what is the next number that you are going to activate? The odd numbers. So then you go for 3. After that, you go for activation of 5. That's a shorter way of learning it. So you start from C1, then you activate the even numbers, that is 2 and 4, and then you go into the odd numbers, that is 3 and 5, and half of your pathway is over. I want to give you obviously a complicated version of it. So let's start. When we start with the classical pathway, guys, obviously there was some antigen antibody complex that started it, and the first thing to get activated was C1, which formed C1q. Okay, QRS, many times it formed. I am interested in C1q. Okay, this C1 then went and activated C4 and C2. Now I just want to tell you one offhand thing. Whenever I'm saying something is getting activated, means it is forming an A version and a B version. Some of the things, some of the A's and B's I'll keep, some of the A's and B's I'll discard. Okay, but when I say something is getting activated, means it's being broken into A and B. So when I'm saying C4 is getting activated, means it's getting activated into A and B. Similarly, C2 is getting activated, it is getting activated into A and B. That's your basic concept. Okay, so remember C4, I want to keep the 4B 2A. So how are they activated? They are activated into C4B2A. So now 4 activated, 2 activated. Even numbers done. Move on to the odd numbers. Which is the one that you will activate? You will want to activate C3. So can I say who is activating C3? 4B and 2A is activating C3. Can I call it the C3 convertase? Can I call it C3 convertase? So C4, that is what was there in the previous one also, if you realized. They were together known as the C3 convertase because they were converting C3 into what? They were converting, again, when I say someone is getting activated, means there's going to be an A and there's going to be a B. This means C3 is now going to combine to this molecule. So what do you have? You have 4B, 2A, 3B. So BAB, alternate chalega. 4B, 2A, 3B is formed. 3 has been converted. 3 is a part of this complex now. After 3, what's the next odd number? The next odd number is C5. So can I say these 4, 2 and 3 that are converting C5 are now known as C5 convertase. And when I say it's getting activated means C5A and C5B, I am now going to leave all the past 4B3, everything I'm going to leave. Now I'm only going to pick up the C5B. And you know what am I going to combine it with? I'm going to bring all its friends together. 6, 7, 8, 9. I'm going to pick up the C5B, combine it with the next 6, 7, 8, 9, and together they are referred to as the membrane attack complex. What is membrane attack complex? Membrane attack complex, whose membrane are you wanting to attack? The bacteria. I want to kill the bacteria. So is this going to go, and for example, if this is a dirty dark bacteria over here that I have made, is this going to go and break the bacteria? Yes, it is going to cause bacterial lysis. Whose membrane is it attacking? The bacteria's membrane it is going to go and attack. It's not a very easy complement system. You read it in weather path or micro, it's tough, but we'll try to simplify it. Let's repeat. The story starts off with C1. It has to start with number 1. First, I'm going to target all the even numbers. Then I'm gradually going to go and target all the odd numbers. So first I activate 4 and 2. So C4B2A is formed. That is now going to activate 3. So then what we have is 4B, 2A, 3B. That is now going to activate 5. And as soon as 5 is activated, 5B, 6, 7, 8, 9 are going to form the membrane attack complex and kill off the bacteria. Okay, having said this, coming back, you realize that your classical pathway is finally a tick mark. Let's talk about the alternate pathway. Alternate pathway is not going to start from that one and all those usual. Alternate means it's going to take some alternate route. It is going to start from C3. That's why it's alternate. It's taken some alternate route. So C3 along with a lot of factors, factor B, factor D, properdin, it is going to break. So please remember, I'll try to fit in the alternate pathway over here only. When I talk about the alternate pathway, the story starts with which antibody? With IgA, and it is going to start with C3. So please remember, C3 is going to be broken into 3A and 3B as always. Every molecule is A and B. So this 3B is then going to combine. This 3B is then going to combine with something known as factor B. Please listen to this very carefully. Factor B, factor D, and properdin. There is involvement of factor B, factor D, and properdin. And you know what happens? Especially factor B. Factor B also starts breaking like these numbers into BA and BB. So you know all the B's come together. I have something known as 3BBB. The 3B from here and BB from here, they come together and they make a complex called 3BBB. And you know whom are they going to go and break? Again the C3. They basically want to come and merge with this pathway at the C3 level. They again go and merge at the level of C3. So first C3 broke. Let's do a recap. First C3 broke into C3B. It took care of all the B's together. It's taken all the B's of its family 3BBB and has gone and attacked another C3. So now ultimately what is going to happen? If it attacks the C3 once again, is it going to result in the formation of 3BBB, 3B once again? If it's again going and attacking the C3, means it is once again going to form C3B, BB, 3B, and finally after that, that will go and attack your C5. And once C5 is attacked, it's going to be the formation of the membrane attack complex. You can have a look at it once again. Let's have a look at it again, guys. We started off our story with what? We started off our story with 3B. 3B acted upon with factor B, factor D, properdin, 3B, BB bana. That has gone and again acted upon on C3. So means C3B, BB, 3B. And once that complex is ready, that is going to go and act on C5. Once C5 is done, I have the membrane attack complex ready. That was the alternate pathway. Now I'm only left with the mannose binding lectin pathway, and that is neither going to start with C1 nor is going to start with C3. If I see, this is starting with the middle molecules and directly going at C3 convertase, means it's starting off with molecules called C2 and C4 and is directly going and targeting where? C3 convertase. So if I realize now which is the meeting point of all these pathways, be it classical or alternate or MBL, they are coming and meeting at the level of this C3 convertase, means they are coming and meeting at the level of something where I want to break C3. And that means what is the most critical step that I can talk about in the complement system? Please remember, the most critical step is a very favorite question, and that is the conversion of C3. The C3 conversion is the most critical step because all of them are going to come and sit down over there. Okay, having said that, I have a couple of questions for all of you, which is based on whatever we have studied up till now. Nothing new, but whatever we have studied up till now, let's see if you are able to answer it. Can you remind me, out of all of these, if I ask you, what is the composition of membrane attack complex? That is what they will be asking you. What is the composition of membrane attack complex? It is C5b to C9. So 5b, 6, 7, 8, 9. Question number 1 sorted. What is the composition? Or what is the next question? Which are all of these which are chemoattractants? If you remember, we had one question on chemoattractants which are going to cause chemoattraction, and one question was there on opsonins, right? So now we need to know those. Please remember, for chemoattraction you had the A molecules, particularly C5A, which is a very famous question. Then what else can the A molecules do? Any idea? What else can C5A, C3A, all these AA molecules, they are also going to be anaphylotoxins. They are also responsible for anaphylaxis. So remember, all the A molecules are anaphylaxis. All the A molecules are attractants, but C5A is the most potent attractant. And all the B molecules, C3b, 4b, 5b, all the B molecules were opsonins, right? Are we clear with this basic? Nothing new that I taught you, everything from what we studied. Now I'm going to teach you something new, because with this complement system, you also need to know all the diseases. So tell me, which is the most common complement deficiency known in humans, and that is C2. May or may not come across as a disease, but at least C2 is the most common complement deficiency. Okay, in someone, if the early complement proteins end up undergoing a deficiency, C1, C2, C4, that has been associated with an autoimmune disorder, that is SLE. That is associated with SLE. So early ones, SLE. If the late ones undergo a defect, that is C5b to C9, C5b to C9 was membrane attack complex, there is infection with many capsulated organisms, and one very famous infection is going to be Neisseria. Let me focus on this, very important, very famous infection that you need to know is Neisseria infection if there is MAC deficiency. Now comes the question that has come so many times in NEET, in FMG, in INI-CET. What is C1 inhibitor? So now, all said and done, everything was going, this pathway was going on and on and on, and in the end, membrane attack complex was forming everywhere, bacteria was being killed. Do you need something that should stop this pathway? Because if this keeps going on in my body 24/7, it will end up killing me only. It will end up killing my own cells. So when required, it should work, and when not required, it should not work. So they realized that you need some people to control this pathway. And that is when they came up with that if you do not let the first step occur, C1 is inhibited, then the entire pathway will not occur. So simple. If you want to control this pathway, do not let the first step occur. So for that, God has given us something called C1 inhibitor. C1 inhibitor controls the activation of C1. It controls the activation of C1. Basically, it controls the complement system. But now I'm talking about a disease when this controller is lost. If the controller is lost, then, like for example, if you are trying to get distracted, I am the one who's going to control you. But if I only go away, or if you switch off this app right now and send me off, then there's no control. Then you will be doing whatever you want. So if the controller is lost, big time problem. And that is going to be hereditary angioedema. That's the name of the disease that you all have to learn. It results in hereditary angioedema. And please guys, the way I've said edema, first thing you need to know, where is the edema going to present? So the edema is most commonly present around the eyes and the lips. So let's draw a funny looking diagram. So if this is a person, for example, and if they ask you where is the edema going to be, so the edema is going to be a weird swollen kind of an eye. Number one, the edema is periorbital, and second, the edema is going to be around the lips. So a very weird looking man that we had drawn, right? So remember, there is a periorbital and a mouth or around the lips. That is where the edema is there. All that is okay. When can this come as a medical emergency? Because that is a question that comes. What if the person ends up having in the neck, means larynx? If someone ends up having laryngeal edema, then this means he will not be able to breathe. If there is laryngeal edema, this means the patient has entered into respiratory distress, and that makes it a medical emergency. Run to the hospital. So remember, if the edema is around the eyes and lips, is still understandable, but if it occurs in the larynx or the respiratory tract, big time medical emergency. That is what has come in the question a lot of times. So remember, hereditary angioedema is because when the controller is lost, C1 inhibitor is deficient. When you're talking about this, why don't you talk about the others also? Let's talk about all of these. And you know what are all of these? All of these are controllers. That is what I want to tell you. All these things that are mentioned over here, CD55, CD59, factor H, factor I, CD46, all of these are controllers of the complement system. So, CD55 and CD59, what do they break? How do they control? They break the two most important odd number converters, C3 converters and C5 converters. So if these three are broken or these two are broken,
complement system will not work. So they're good for us. CD55 and CD59 control your system. But just imagine if CD55 and CD59 go missing, the controllers are gone. Does it remind you of any disease in the blood? There is a disease called PNH, which means there is paroxysmal nocturnal hemoglobinuria. There is paroxysmal nocturnal hemoglobinuria that you have to know. Okay, next let's look at factor H, factor I and CD46. They are also controllers, but when factor H, factor I and CD46 go missing, it's a very important disease in medicine and pediatrics. That is going to result in atypical hemolytic uremic syndrome. Atypical hemolytic uremic syndrome. Finally, I want to correlate this with ophthal also. Have you guys studied that factor H is associated with something? Factor H defect in ophthal, what can you think of? Have you heard of ARMD, that is age-related macular degeneration? Age-related macular degeneration, that is because of factor H defect. So this is basically trying to tell you all the relations of complement systems that are going to come in the future in your life, be it in path or be it in other subjects. So remember, CD55 and 59 are PNH, factor H, factor I and CD46 are atypical HUS, factor H in ophthal is age-related macular degeneration. And for the time being, you just need to know that the same disease I will be telling you under, or the same set of things I will be telling you under kidney, because now there is a disease called MPGN, which you have to know, which is very, very important. So please remember, for the time being, I can only tell you a little bit about it. MPGN type 2 is now said to be a complementopathy. It is said to be a complementopathy, means there is a problem in the complement system. What is MPGN? It's a disease in the kidney, membranoproliferative glomerulonephropathy. Membranoproliferative glomerulonephropathy is a defect in the kidney. So right now just know it's something to do with complement system, okay? Having said that, the complement system is done.
And that finally marks the end of your entire immunology chapter. So like I said, everything of path and micro was same, but this section of immunology was more important in microbiology. If you feel thoda sa confusion hai jo rahega, after this you're supposed to go to the RR videos and jaldi say, I'm going to make a table over there, and over there that will be a very rapid review of all these precipitation, agglutination, liquid, gel. So there's going to be a very concise version of that. So next time whenever you want to revise, that's your go-to thing.